Welcome back, class. Today we're going to cover just osmosis. What is osmosis? Osmosis is just the diffusion specifically of water across that selectively permeable membrane. So look at that bottom picture. On the left-hand side, you have a hypotonic solution. I'll go over these vocab later on. And you have a hypertonic solution. If you look at that hypotonic solution, it's got very few molecules suspended in water, which means that there's a very high concentration of water on that side versus the right side, there's a lot of those random molecules and there's very little water. What's going to actually happen is water is going to go across that selectively permeable membrane to the right. It's going to go from a high concentration to a low concentration. So you're going to see that water level on the right-hand side go up. And you're going to see the distribution between water and purple dots to be equal on both sides. So, cells and osmosis. If two solutions are separated by a selectively permeable membrane, we're actually just going to move those water molecules across that membrane until they are equal concentration or in equilibrium with each of the sides. That kind of explains the image that we were just looking at right here. Moving forward, cells are often surrounded by water or water solutions or substances that are very high in water concentration. Depending on the solution concentration, the cell's shape may change. What do I mean by that? We have three options actually. We have isotonic solution, hypotonic solution, hypertonic solution. We're going to go through all of them. But osmosis in cells depends on what type of solution those cells are soaked in. Okay, think of humans. What kind of solution is our body cells suspended in? Okay, think about it. Think. Hopefully, you guys are thinking about all that blood that flows through us, and we're in the 90 percentile range of having 90 or 95 or 96% more percent of water in our body. So isotonic, iso, think of isosceles triangle, equal sides. So water concentration in the solution is equal to the water concentration inside the cell. So water moves in and out of the cell at an actual same rate. And we call this dynamic equilibrium because it's constantly fluctuating back and forth. But it is remaining in equilibrium, okay? The cell, we have a red blood cell on the top and a plant cell on the bottom, is going to remain a normal shape. Hypotonic, very slow. Okay, hypo means slow, means the opposite of hyper, which is the next one. The water concentration in the solution is greater than the water concentration inside the cell. So water is going to end up rushing into the cell, but let's say we put a red blood cell in distilled water, which has a very high concentration of water molecules with no minerals or salts added to it, like spring water. The cell, water is going to rush into the cell, and because the cell, an animal cell, doesn't really have a very big structure, it's going to burst, like you see that red blood cell on the top. Looks like a balloon that just burst. Versus cells inside a plant cell, they're just going to become swollen, okay? And that process of being swollen is being kept from exploding because plant cells have cell walls. They're very rigid. They help with support and structure. Then lastly, you have hypertonic. Think of someone who's hyper, very fast, it got very high. So water concentration in the solution is less than the water concentration inside the cell. So think of it, if I put my cell in salt water, what's going to happen? There's very low concentration of water outside or surrounding the cell. So therefore the water inside the cell is going to move out because it wants to go from high to low. So what's bad about this for an animal cell, you're going to see things get very shriveled up, okay? But with the plant cell, it's really interesting. You're actually going to see the cell membrane separate from the cell wall. We're actually going to do this later on in the year with one of the state labs. I hope this lecture helped you understand the difference between isotonic, hypertonic, and hypotonic solutions and apply them to the cells. If you have any questions, don't forget to ask me in class.